to help bridge this gap between
live streaming is on.
activated sludge microbial science and wastewater engineering practice. I hope you got my introduction, otherwise. But before I carry on, I'd just like to very briefly introduce the company I work for, Orvion, to you. Orvion is an environmental biotechnology company located in the Netherlands. And ever since it was founded in 2013, its primary focus really has been applying naturally occurring microbial processes in environmental practice, for example, for groundwater remediation or wastewater treatment. Because we work so closely with these complex microbial communities, from the beginning, we've also always been working with DNA techniques such as Qpcr and next generation sequencing to be able to monitor these processes, control them, and understand what they're doing in practice in the environment. This has also recently resulted in the development of a mobile Qpcr tool, whereby we really aim to provide Qpcr monitoring for specialists and non-specialists wherever it might be needed. And as I'm sure you all know, if you're working with NGS and Qpcr, there's a lot of data that's generated. Same for us, we've really recognized that it's important to develop bio-IT and software applications to be able to handle this data, to be able to make it visible and understandable, and especially for the clients we work for and with, who very often have no microbial background whatsoever. And I'd just like to briefly touch on an environmental sector where this microbial data and microbial monitoring has already been used in practice for at least the past 15 years. And that's for bioremediation of groundwater contaminants. And this is a sector where you work together closely with governmental authorities who have to approve of remediation projects before they're allowed to be performed. We're working together with engineers and contractors who are actually the ones that perform the work that needs to be done. But essentially, the processes they work with are going to be biological. So there also has to be a great degree of biological microbial knowledge that's integrated in these projects. And it was very quickly realized that in addition to the chemical and physical analyses of the groundwater and monitoring of groundwater processes occurring, it was valuable to also really monitor and understand the microbial processes occurring. And to do this, there was a very good collaboration with scientific research that was happening in this area, with environmental consultants and experts who knew how to interpret and analyze the data, and the contractors and authorities at the end who were going to actually use this information. So for example, I think as early as 2004, 2005, it was started to use Qpcr for just routine monitoring of biodegrading bacteria in groundwater samples of a contaminated site. And of course, as research progresses, there's new microorganisms being identified that have capabilities for biodegrading contaminants and the genes that they use to be able to do this. And now it's very routine, really, to analyze groundwater samples using Qpcr for different biodegrading organisms and genes. And this is just an example of a way that they can get information on where in the site is the potential for biodegradation already present, where is it maybe not present and you really need to get stimulated. But also once you have started remediation processes, it's also a way to monitor whether their numbers are increasing over time, so whether the remediation is going according to plan. Increasingly also, next generation is being used for these types of projects, especially for more complex contaminated sites, or when there are so many organisms now known that are able to degrade these contaminants that using Qpcr doesn't become a very viable option. And this is just one example I wanted to show you, if I can find my pointer. There we go. Of a site that's heavily degraded with naphthalene, and I hope you can see the lines clearly. This is the naphthalene concentrations in different wells in one area of the site. So these are different areas that were analyzed. And what immediately becomes clear from this data and how it's presented is that in the higher concentrations, there's a great dominance of a bacterial genus called Desulfitobacterium which is known to be able to degrade this component. And also combining, of course, the other data that's available from the site, it becomes very clear that in these areas where there is a high contaminant, there's also a lower degree of the electron acceptor that's required to degrade the component, being sulfate. So you can clearly see that in these areas, the sulfate concentrations are lower, as shown in the blue lines that are visible. And basically what this shows is that, yes, there's a great potential for bioremediation at this site, but it might be beneficial to actually stimulate it by actively adding more sulfates to the site in order to promote biodegradation processes and make them go faster. And just being able to show this data to the authorities and to the contractors has actually now resulted in a pilot project being started just last month, whereby they are actively going to stimulate this biodegradation by adding sulfate. So this is just an example I wanted to show you of how this kind of information can be used in practice for decision-making purposes, but also to really clearly show also the non-experts how it can be of value. So how can we do the same in the wastewater treatment plant world? Now, this is a picture that's already been shown, but I think it very clearly illustrates one of the questions of how can we get the data and the information that's generated in the lab and make it useful for the operator who's standing in the plant and has to make decisions on how to run it. And vice versa, it mustn't be forgotten that there's a lot of data information being generated in practice by the wastewater treatment plant that can be used again for scientific research purposes. And I think one of the keys is really thinking of how we can translate the information from both worlds so that they understand each other better and can work together better and can really start to use the data effectively in practice. Now, in the past years, we've been investigating exactly these questions together with a number of our Dutch water boards to see how we can really get to grips with this challenge. And luckily in the Netherlands, and apparently quite uniquely in the Netherlands, our Dutch water boards who manage the wastewater treatment plants in the Netherlands are highly involved in innovation, seeking to start up new innovation, actually actively participating and financing this kind of innovative research, so that the framework is really present for this sort of stuff to actually happen. And interestingly, also in the past few years, there's been the DNA techniques and how they can generate information has become a very relevant topic to water boards, to see how they can use it for additional data generation, monitoring, and how it can be of value to them. And this was really a good sort of starting point to work together with them. And so in the past few years, we've performed a number of projects together with them to see how using DNA techniques and starting to really understand and make the microbial community visible can be used in practice. So in the past few years, we've been able to monitor 3 municipal wastewater treatment plants during a period of 12 to 15 months, as well as 2 industrial wastewater treatment plants. And there's also been a number of plants that we analyzed just once or a couple of times owing to a calamity or something that happened there, whereby the water board is interested to see what's going on and what effect has this had on the activated sludge. Now, all the analysis we performed during these projects was on total DNA, so there was no pre-amplification, and we always used nanopore sequencing. And part of the projects, we also focused on seeing which topics are really of interest that we could really start to focus on. And I think in general, there's a lot of interest just to get an idea of the different trends in biodiversity that are occurring in the activated sludge over time, but also being able to start to compare different treatment plants. There always seems to be one that performs a bit better than the other in one way, and vice versa, the other seems to perform a bit better than the one. And if you can see what the differences are also in the microbial community, you can start to maybe better understand why these differences are occurring and how you can then learn from each other. Other topics probably very familiar to you all, the pool sludge settlability owing to, for example, filamentous bacteria, biological nitrogen removal, especially, for example, nitrification that can get inhibited, phosphorus removal, so the enhanced biological phosphorus removal, Recently, there's a lot of interest for preventing nitrous oxide emissions, which of course also has a strong biological component. And also interesting, one thing that always comes back is being able to source or track the source of a toxic discharge that may be occurring from industry. So who's the one that might be responsible for actually causing nitrification to be inhibited? So these are some of the topics that seem to come back very regularly and that we really started to focus on. So as I'm sure you will recognize, once you start this kind of project, the first thing that really happens is that you're overwhelmed with the amount of data that you're actually generating. So per sample, there's a lot of information coming away about the microbial community, but of course also centrally it was very important for us to get all the process data from each of the plants we were monitoring and integrate that with the microbial community. And to do this, a lot of bio-IT and IT programming was occurring. But if you want to make this data usable, you need to make it user-friendly, as I think most people are still quite terrified when they see sort of Python scripts on their screen that they have to use. At least I am. So it became very important to be able to sort of combine all of this information in a central way that's usable, not only for wastewater treatment plant operators and engineers, but also for the scientists that want to look at the data in more detail. And that's how we really have recently developed our activated sludge platform, whereby we're trying to get this happening. So here, just briefly, how it works. On the one hand, the activated sludge platform is really centrally, it's the data that's key, of course. So on the one hand, we're adding all the microbial monitoring data that's occurring from each of the plants and combining that with the process data that the water boards share with us. But importantly, what we also really want to start doing is to combine this with data that's being generated from scientific research. And this can be very basic primary data, but just to get it included in this platform. And then there's different users that might be interested in looking at the data that we're generating. On the one hand, simply a view of how your plant is operating, how it's changing. So just getting insight in the plant key performance indicators, also including your microbial community. On the other hand, for scientific research, I mean, there's a wealth of information here also from practice, all this process data, all this microbial monitoring data that can be used to further scientific research. And of course, wastewater treatment plant engineering that also want to learn and get the data so that they can actually start making decisions based also on the microbial community of the wastewater treatment plants. And importantly, to help support some of the decision-making challenges that they face, just having a complete set of data. So in the next few slides, I just want to show you some of the tools we've already integrated in our platform, what they look like, and some of the data that we're coming across. So in this first screen, what you really see is just some of the basic, perhaps key performance indicators of a plant. So on the left, you can select which plant you want to look at, depending on which water board you are, I suppose. And then just very simply, just by selecting different parameters you want to have visualized, just see some of the processes that are occurring. These are all just stuff that they monitor anyway, but this is nice because it's integrated here anyway, so you get an idea of what's going on. So for example, your CD in the influent versus effluent, so how effectively are you removing things? Something like the sludge volume index to see how your settlability is, or something simple like just the temperature in your tank, which of course for a lot of microbial processes is quite key. So you want to be able to look into that. Now moving forward to the more microbial information, this is just two different ways in which we visualize the microbial community that we find in our activated sludge. So on the left here you have a heat map visualization. And in this heat map, the data has been filtered for filamentous bacteria. So there's different filters defined for nitrification or for the phosphate accumulating organisms. So you can actually filter the data as you like. And you can also regard it at different taxonomic levels. So this is for genus, but you could look at family or whichever level you're interested in. So this is just a nice visualization that's often also used in articles just to visualize how the population is changing in time. And on the right-hand side is actually exactly the same data, but just visualized in a different way. We have the stacked bar charts, once again filtered for filamentous bacteria over time. And it's very easy here on the right to toggle between different organisms you want to visualize just by selecting or deselecting them. And in this visualization, it also becomes possible to show the process parameters in the same view. So at the top, you can just add whichever process parameter you want to have visualized. So in this instance, it's a sludge volume index. So this is becoming already, generally it's very understandable, but it's starting to dig deeper into the microbial community. But of course, if you're scientifically interested in what's going on, you also want to be able to start to look for correlations that might be occurring between the different parameters. So in a different view in the platform, you start to be able to build these correlation heat maps. And it's very easy here at the top to select or deselect various parameters that you want to have visualized, as well as the organisms that have been identified in your sludge. I'm sorry if my voice sometimes goes. I'm recovering from a cold, so I hope it's going to last. And in this way, you start to build this correlation heat map, whereby, of course, the darker colors signify a more a higher correlation between the parameters, being positive correlation or a negative correlation, and it becomes very visual as to which parameters are showing them. And the nice thing is there's a hover over function, so as soon as you hover over one of the correlations, which unfortunately I can't show to you live because this is only running on our local servers at the moment, but you immediately get to see the details of this correlation. So which parameters they are, what the correlation coefficient is, as well as the p value. And immediately on the right-hand side is also a visualization of the correlation. So often you want to see exactly what the correlation is based on. So in this one, it's a relatively simple correlation between this bacterial genus Promineophyllum with the temperature in the tank. And apparently this one prefers higher temperatures if you just look at this correlation. Now, another correlation that's quite high is between this microbial genus Microtrix, which is a very well-known filamentous bacteria, which shows a high correlation with the nitrous oxide emissions that were monitored with a sensor in the same period. So we're not actually looking at the whys and the wherefores, we're just simply showing that there is a correlation present here. And this, of course, provides input for further scientific research, should that be necessary. I'm just going to have a sip of water. This visualization is just a more simple way of showing the data in the same graph. So it's the same Microtrix data shown here in the bar chart, together with the tank temperature, and once again, the nitrous oxide emissions that were monitored. So this just shows different ways that you can visualize the data. And here, once again, it's very easy to toggle between different parameters you want to have visualized. So how could this possibly be used then for sort of daily wastewater engineering practice? Well, what we foresee really is that by making the data and the trends visible for wastewater treatment plants, it becomes possible just to monitor what's happening with some of the organisms that are of interest within a certain topic. So, for example, your filamentous bacteria, how are they evolving over time? And eventually, if you can set a threshold value above when you know that they start to perhaps form problems, you can also start to link actionable data to it. So if it crosses this threshold, it means that then numbers are probably increasing and you need to take action. So it becomes actionable data that can be used in practice. On the other hand, it also can help with diagnosing when you're seeing that your wastewater treatment plant perhaps isn't functioning as you'd like it. Apparently, it's more difficult than I ever thought to really understand when a plant is having problems just based on the physical chemical monitoring, because it does tend to vary quite a lot. So when is it really a problem, or when is it perhaps just a variation owing to your influent quality? And this is something we actually came across with one of the treatment plants we were monitoring. We followed it during 12 months, during a 12-month period in which they went for nitrification purposes. That's what we're showing here, the ammonium oxidizer nitrosomonas is being shown in this plot. And during this period, there wasn't really any calamity with nitrification until about a year later, when they did have a calamity. And immediately the question arose, can we see this in the activated sludge, that there is actually something wrong with our nitrifiers? So we analyzed the sample, and what we saw is that for a warm July month, whereby you usually would have percentages around 1.5–3%, or some nitrosomonas is one of the lowest that we had monitored so far. So this could really help in the decision-making if you can quickly get data on the quantities of your nitrifiers in the activated sludge to say, we do in fact have a problem. And that combined with the physical and the chemical and the operator parameters, just the data that you have already of your plant, to say, yes, we have a problem, and then much quicker start to take action, basically. So this is something we're looking into to see if we can really get that into daily practice. So what's next? Of course, we want to develop our platform further to integrate further Bio-AT tools, but also really for the wastewater treatment plant to get these threshold values, to get the trends visible, and to be able to tell them when and how they need to act. And in this way, hopefully also really start to work together with the wastewater treatment plants and the science that's happening in the universities. And very concretely, within a few weeks, we're going to start a pilot whereby Microtrix specifically is going to be monitored over a longer period in 3 susceptible wastewater treatment plants. So they always seem to have problems with bulking and foaming. So we're going to monitor them with Qpcr to get quick, actionable data, and hopefully also an early warning system so that we can much quicker see that their numbers are increasing. And what the Water Board really aims to achieve is that they can then take action quicker and hopefully also prevent chemical additions being required to get settleability under control again. So that's starting soon, so we're excited to see how that's going to work out. And also what we want to do in the next year is really start a collaboration between a number of the Dutch water boards and the TU Delft to start filling and using this activated sludge platform in practice, whereby we really want to get the data from science to the wastewater treatment plant and vice versa, enabling the data from the wastewater treatment plant to be used by the scientists. So that's something we're hoping to start up next year. And of course, as I mentioned, we generate a lot of data. We have all this metagenomic data available to us. And we know that we're only scratching the surface of all the data we have already. So what we also really want to start doing is providing this information that we have to further scientific research. And that's really what I wanted to tell you today. So all that rests is to thank you very much for your attention. And I'd also very specifically like to thank Fridtjof Gottschalk and Ramon van der Zwaan, who actually do all the data work and all the IT work. So I wouldn't have much to tell you if it weren't for them. So thank you very much.
Thank you so much, Aleida. And I can hear online also participants applause. So it means that the platform works. If you experience any issue with the platform, just write in the chat openly and we will try to solve. It seems that you made quite an effect with your dashboard. Yeah, you can see also the hands going up. It's very nice. It's more than a marriage. Huh? So congratulations for your dashboard, Aleida, Fridtjof, and it's also nice to see how an intern of the university can make a nice contribution to your work. We have time for 1-2 questions. So I don't know if Sergio, you see some question in the Q&A or in the chat. So
there is one question in the Q&A, but also many people raised their hand, but I don't know how to check that. So everyone can write the questions in the chat and we can come back to that and see, we'll check them later. But there is,
select one.
Yeah, the one that it's in the chat by Dr. Niti Jadeja. And in your experience and interactions with the wastewater process consultants, contractors, have they been interested in depth microbiome analysis only during the problem-solving troubleshooting, or did you observe readiness for making microbial analysis routines like monitoring of physiochemical parameters?
That's a very good question. Thank you very much. I think when we started on this crusade, what is it, 5 years ago, I think, there was a higher degree of skepsis, perhaps, especially sort of questions, you know, how can we use the data? What's in it for us? What's it going to cost? So more that aspect. But I think in the past years, you're really starting to notice that people, you know, data is key. If you want to understand what you're doing and optimize what you're doing, you're going to have more data. So to be honest, in the last few years, there's been a lot more interest in not only monitoring when there's a problem, but really understanding that if you don't have a frame of reference for your own treatment plant, you can't really tell if you just analyze it once whether it's okay or not. So it's definitely coming, and that's exactly what we're noticing now. Why we want to carry on with this next year and make it more structural is that the interest is definitely there. But it depends on your treatment plant. I mean, some treatment plants just maybe don't have any problems, or the necessity isn't really there. So it depends a lot on your plant. But the interest is definitely there.
And is there a question in the room as well, so to keep a balance between online and on-site? Is there a direct question? So maybe a question from me. So how do you convince practitioners, because you are directly at the interface with them, that it's the way to go with the dashboard?
I think by working together. I don't think if you have to only convince people to do something, it's never going to happen. So it has to be a collaboration whereby you both believe that it's worth it, and that's what we're really noticing is that people from the wastewater community are saying we're not entirely sure yet how it's going to benefit us, but we do believe it will, and that's the best you can hope for, and progress from there.
Thank you very much, Aleida. And again, a round of applause for Aleida. And we will move next to the sessions of oral presentations. We will have 5 speakers, and Sergio and I will co-moderate that. So please, Sergio.
So now we are going to go with the pre-record, and the next speaker is going to be Susan Hansen from Aalborg University, and we are going to launch the pre-recorded presentation. And the presentation is ozonation for removal of micropollutants creates positive effects on the microbiology and wastewater treatment plant performance.
Hello, everyone. I am Susan Hansen. I'm a postdoc at the Center for Microbial Communities at Aalborg University in Denmark. And I'm about to present to you a very nice project about ozonation in a full-scale treatment plant in order to remove micropollutants. But also, I'm going to tell you about some of the positive effects we saw on the microbiology and the process performance. So, this project was a big collaboration between the engineers and industrial partners, and it was mainly sponsored by the Danish Environmental Agency. And we were so lucky that we found this little plant in Denmark with 2 parallel lines that was very suitable for this system, for this test. And one line was used for ozonation, the experimental line, and then the others kept as a control line, so we can always compare what happened. And the setup was that the ozone was put directly into the process tanks, and the effect of that was actually very positive. So we saw that up to 90% of the pharmaceuticals were removed, and also we saw that the sludge actually settled much better. And the 2 lines actually seem to work in a similar way, so they had the same biological performance for removal of carbon and nitrogen and so on. So that was all very nice, but of course as we were partners here, we of course wanted to ask, what about the microbiology? How is that affected by this ozonation? So we asked the following questions: that were the two lines comparable in terms of microbial communities before we started the ozonation testing? And also, were there any effect of ozone on the microbial community? And if yes, which bacteria were then affected? And could these bacteria then be correlated to these improved sludge characteristics that we saw? And I can show you here what we saw from just looking into the microscope, that here we could already see some effects of the ozone. Some of this attached growth that we see on the filaments in the control line, they were gone when ozonation was on in the process tank. So that was a first glance. But to get even more detailed look into the microbial community, we did the 16S RNA amplicon sequencing. So the engineer at the treatment site, he sampled every week the two lines for more than a year. After that, they sent all the samples to us, and we extracted DNA. We prepared all the samples, and we did the sequencing. And then we used the Midas taxonomy in order to get some names for these sequences. And the result we get is this long list of bacteria, and then in which abundance we see them. And I can here use a little time to advertise then for my smart colleague Morten, because the reason we can use and go to this very high resolution is because we have this Midas 4 taxonomy now. And Morten, he will tell much more about it on Tuesday, how he did that and how he developed this. It's already cool. So let me come back to the result from this project here. This is actually how it looks like when we present the microbial data in heat map format. So what we see here on the y-axis is the names of different bacteria. And here on the x-axis, you have the time, more than a year. So each column represents one weekly sample. And the more red, the more bacteria, the more high abundance, and the more blue, the lower abundance. And we can already hear from this heat map see that some bacteria seem to like the ozonation, and some seem to disappear almost. And I can also tell you that the way it worked in the whole year of sampling and testing was that half a year was spent just simply to make sure the baseline was established, and we have monitored that. Then 3 months of ozonation, and then after that, actually 3 more months with the double dose of ozone was performed and tested. So this is a little bit complicated to look at. So we always like to do this ordination with this type of data, because then we can see more structures and development in the microbial communities. So when we do ordination, we actually condense all this information from one single column from a heat map into one point in such a plot here. So the more similar 2 points are in the plot, the more similar the microbial community are. So what we could see here from this plot is that the two lines were very similar in the baseline. So the dots are very close to each other in the test line and in the control line. But then when ozonation was started, the two lines seemed to drift apart and start to differ in the microbial composition. What we also saw when we went down to species level and put some threshold of that, if we look into a species that is at least seen twice in an abundance above 0.1 over this whole time period, we saw around 500 species in the process tanks, and out of them, actually 328 was affected by the ozonation. And we can also show that the two levels of ozone dosage also affected. So the more the ozone, the more bacteria or more species were affected compared to the control line. And then we looked into these the functional groups of the known bacteria that we know that performs the nitrification. So here we took a look at the Nitrospira and Nitrosomonas. Here we look at the genus level. But what we saw was actually very nice, that they seem to like it under ozonation because they increased in relative abundance. And that's of course very nice. And as I also mentioned before, we have screened many of these functional groups and we actually didn't lose any of them. So this is how it looked. And also with the -4 taxonomy, we're actually able to zoom even deeper in and look at the species level. So what we saw, very interesting for Nitrospira, was that one of the species, with this specific Midas name here, species 361, seemed to thrive under ozonation, whereas the other one, more well-known, Nitrospira diffluvi, seemed to not be affected very much compared to control line. So that's nice that we can now go into this detail level. Another thing that we saw and what we looked into was the filamentous organism. And here we have a few Chloroflexi genera here, the Vilicacilis and this Ambulinia, but we can see that they seem not to like the ozone at all because they almost disappear, especially with the high doses. And unfortunately, in this specific plant, we had none. We didn't see this well-known troublemaker Microtrix that we always have the troubles in Denmark, Danish plant especially, but unfortunately it was not there. It would have been nice. But what we did saw was that the sludge settling improved a lot during ozonation. So if we measure this SVI, sludge volume index, it drops drastically under the ozone treatment. And we hypothesized that this could have something to do with the filamentous organism. As we here, if we look at the list of the most abundant filament species we found, we have actually a few of them showing a significant response to the ozone. So it was tested here if the two lines were very or significantly differed before or after ozonation. So we found some that did, and yeah, that leads to the end of my talk. So I would like to sum up with some conclusions. So this full-scale demonstration of micropollutant removal by ozonation was actually very successful. We removed the micropollutants, and the plant seemed to perform as it did without ozonation. So that was all good. And with our specific inputs from these microbial data, we could actually put some more power to the conclusions and aid some chapters in this report we had to deliver to the environmental agency. So we could actually confirm that the two lines had similar microbial communities. So it was a very good choice of treatment plan to test in full scale for such an experiment here. And also that this DNA-based method, they could actually back up the biological performance data that we didn't lose any of the critical organism in the plant when we ozonated. Then we found that the nitrophiles actually thrive under ozonation, and that we actually saw a little bit of differences at the species level here. Also, some that didn't thrive very well was some of the filamentous organisms, but they actually, the loss of some of these seemed to correlate with this improved settling properties. That's always good to have. And again, here we see definitely some differences at the species level too. So that was it from me. Thank you for your attention, and thanks to all my colleagues in Aalborg and the partners of the project. Thank you.
Thank you, Susan, for the presentation. And I think I saw you online, so we have time for a short question that you can answer now. David, you asked the question.
So if Susan, you can join online. Christina Galliano from Vetsus in the Netherlands has a question about any idea about why ozonation is stimulating the growth of NOB and AOB.
Hi.
Hi, Susan. We can hear you.
Well, we have been discussing that a little bit back and forth, but I'm not sure. But I think there must, because of the ozonation, there will be also extra ozone available maybe, and that's something that can that these nitrophiles seem to like. Yeah,
actually, there are quite some avenues to try to delineate why there is this ozonation or chemical interaction with the biological populations that are present. And understanding the mechanism behind will be nice in the future. Thank you so much, Suzanne. And please stay on. Mark has a quick question. You can go at the microphone, and then we move to the next pre-recording. Yeah. Can you help, maybe?
Where's it's red, so it should be on. Yes, it's on. Just a question, because if you see an increase in number of, say, nitrifiers, is that related to the fact that you have decreased other organisms and it's just a relative increase, or it was an absolute increase?
Yes, thank you, Mark. Well, it's of course a relative increase, so it can also be that many others seem to die off. But we thought, since they don't disappear, it's very positive trend. But others seem to be stable over the time. So
yeah, this brings always the question about units, how to combine relative abundances and absolute values. It's clear ampli sequencing does not give absolute values, but maybe we can still multiply the relative abundance times the VSS to assess how much organisms there is roughly. Yeah. Thank you so much, Suzanne, and stay online for the breakout session afterward. We move to the next speaker, Sergio. Thank you.
So our next speaker is Benjamin Ginkley from Northeastern University, and he's going to present autonomous in situ monitoring of complex microalgal communities, also pre-recorded.
Hello, and thank you for joining me to discuss autonomous in situ monitoring of complex microalgal communities. I would like to thank the International Water Association and the organizers of this panel. My name is Benjamin Ginsley, and I'm a graduate student from the US at Northeastern University in Boston, Massachusetts. I'll present a novel, low-cost imaging-based platform we've developed to address a current technological gap in the market, as well as describe some of the application areas we're currently exploring. I invite you to ask questions about the design or use of this device at the end. The problem as old as civilization: nutrient pollution of the environment. When nutrient-rich discharge or runoff is received by surface waters, microorganisms already present in the environment capitalize on this fertilizer and rapidly grow in what is described as an algae bloom. These blooms can disrupt normal ecosystem function, cause discoloration or foul odor, and depending on the type of algal species present, can release toxins that are harmful to animals and humans. Such harmful algal blooms cause significant economic damage on their own and require further management to mitigate their effects. One such management requirement is the careful monitoring of receiving waters to detect when an algal bloom is imminent, to inform proactive action that can prevent a full-scale bloom event. As a testament to human ingenuity, we've begun to turn this problem into a solution: controlled algal blooms and engineered systems that remove nutrients from wastewater before it is discharged. This minimizes eutrophication of surface waters and also provides a renewable source of biomass that can act as a fossil fuel replacement for valuable co-product generation, including biofoams, bioplastics, inks and dyes, and biofuels. To optimize growth in these engineered systems, the microalgal community requires monitoring to enable closed-loop control of process parameters. Therefore, there exists a need for affordable and automated monitoring technologies that can operate both in engineered systems and in the environment to monitor growth and changes in microalgal community structure. Deciding on the right approach to quantify individual constituents in the microalgal community requires consideration of multiple requirement dimensions. What are the capital and operational expenditure budgets? How quickly does the sensor need to provide results? How detailed and accurate does the data need to be actionable? Different approaches are optimized for different facets. Probes can provide data very quickly, but can only report on metrics such as total chlorophyll or total suspended solids, and don't provide insight into community structure. Nucleic acid sequencing methods can provide the greatest level of detail, but are expensive to automate for field use or require a high level of technical expertise for good extractions. Sending a sample to a facility can take days or weeks to return results. Flow cytometers are very fast at processing samples, but are also often prohibitively expensive for a distributed monitoring schema and require samples to be collected from multiple points and sent to a centralized location, precluding autonomous online monitoring. Here we present a solution that can provide a high level of detail at very low cost compared to comparable technologies currently on the market. That solution we call Artemis, the Autonomous Real-Time Microbial Scope. This is a compact and lightweight device that can fit in a backpack and can run from a 12-volt battery. It has been designed with simple operation and maintenance in mind. No dyes or stains are required for high contrast images, and both the fluidic and optical components can be set up and swapped quickly. The core design has a low component cost relative to similar products on the market, thanks to the creative use of some non-traditional optics. The device body is currently 3D printed to facilitate rapid prototyping to keep costs low. I'll also note that the platform has been designed with modularity in mind, with the ability to swap lenses, cameras, or flow cells depending on the application. For bright field, dark field, or quantitative phase imaging, algae and suspension go directly into the device. Once images are captured, they can then be processed in a number of ways, including traditional object detection and classification analyses, and I'm excited to go beyond object classification in the future. This device targets low resource contexts, and I don't only mean developing economies. This also includes deployment locations where it's infeasible, impractical, or out of budget to have full-scale lab equipment at the point of sampling, and expert personnel on site to prepare samples. To operate the device, we have developed both a graphical user interface as well as a programming API for scripted processing on device startup. Here are a few example images to demonstrate some of the application areas we've explored so far. On the left, we've been using this platform to track changes in community structure at Clearest Water Recovery's EcoRecover wastewater treatment process in Roberts, Wisconsin. We can use this image data to correlate and later predict how substantial changes in influent wastewater impacts the algal community, and inform how that may impact process performance metrics like phosphorus uptake. We've also worked with environmental water quality researchers in New England to automate phytoplankton counts to track and predict surface water algal blooms. We have demonstrated submicron resolution with this optical system. On the left is an image of a resolution test target used to determine horizontal and vertical resolution. The smallest element line pairs, group 9 element 3, are resolvable in computational analysis, suggesting a resolution limit at 0.79 microns. Below, images of E. coli cells in 0.5 micron beads, demonstrating resolution of micron and submicron scale particles in solution. We also demonstrate accurate density reporting across a range of loaded sample densities. Calibration microspheres were serially diluted and processed on the device. At higher densities, an accurate estimate converges in less than 5 images, or about 3 minutes. At lower densities, it can take longer to encounter enough events to have an accurate estimate, but convergence usually occurs in under 15 minutes. As this is a flow-based system, it can be run for longer time to increase accuracy, especially at low densities. In the sample processing pipeline, after image capture comes image processing, which is done on the embedded single-board computer. On a preliminary and relatively small initial training dataset, we've seen high accuracy when distinguishing between 3 common microalgal genera. I expect this performance to improve as the training set size increases. On the right is an example of this model deployed to analyze a pure Chlamydomonas culture. Green boxes represent Chlamydomonas predictions, purple for debris, and red for sedativeness. This is one of the things that never ceases to amaze me about this project and what's possible with modern deep learning techniques. With a simple yet carefully designed model, a high-quality data set, and just the right amount of dimensional reduction, you can get highly accurate classification on a computer the size of a credit card. For those who are familiar with popular deep learning models deployed on desktop or the cloud, including AlexNet and ResNet, this model has 40,000 parameters versus the tens of millions of parameters in such models, which enables it to run on embedded systems. We saw a snapshot earlier, but we are currently testing this with our industry partner, Clearest Water Recovery, where we are part of a joint effort funded by the Department of Energy. A key output goal of this project is to inform when and how operators should adjust plant parameters to 1, maximize phosphorus uptake to meet regulation limits, and 2, maximize biomass yield. While this site is highly instrumented with nutrient and water quality parameter sensors, instruments to continuously monitor the algal community directly are harder to come by. TSS probes alone can give a good estimate of total biomass, but the readings may be hard to act on when you're trying to maximize macronutrient levels for a specific co-product. We're beginning a collaboration with Oak Ridge National Lab to go beyond object classification to see if we can use techniques like deep learning to measure attributes like lipid accumulation. We're also developing partnerships on the surface water side with the Great Lakes Water Authority to explore tracking and predicting algal blooms and their impact on drinking water sources. I'd like to acknowledge and thank the funding sources that have enabled me to develop and continue to advance this project. I would like to thank the John J. Cochran Fellowship Fund, the Water Research Foundation, the National Science Foundation's Graduate Research Fellowship Program, and the Department of Energy. I'd like to thank my collaborators at Clearest Water Recovery and Oak Ridge National Lab, as well as our academic partners at the University of Illinois and University of Buffalo. I'd also like to thank my primary research advisor, Dr. Ami Pinto, as well as the graduate and undergraduate students that I had the opportunity to work with while developing this platform. Last, I'd like to thank the organizers of this session, and thank you all for your time and attention. I'll be happy to answer any questions in the allotted Q&A period and from my email address on this screen. Thank you.
Thank you very much, Benjamin. Are you online? If you are online,
Yes.
you can unmute your microphone. Is there a question from the room about this microalgal monitoring? I have one question. Then what is the time to action between the measurement that you make in the water system and the action that the authorities or the wastewater treatment plants or the process engineers can apply to minimize the impact, for instance, of nutrients on the eutrophication?
Yeah, that's a great question. I think first off, the time to results for a device, depending on the concentration of microalgae in the system. For example, if we're looking at receiving waters, we can typically get results within a few hours, and from there, it's kind of up to the authorities to decide what method of treatment they're going to do. If they're doing a chemical treatment in the receiving waters, they can send a boat out to do chemical treatment maybe that same day. If they're trying a more proactive, preventative approach on reducing nutrients that are entering the system from runoff of like phosphorus and nitrogen and that sort of thing, obviously the time scale of how they're going to act is a little bit different. But in terms of the device returning results, you can act on the same day.
Thank you so much, and stay online for the breakout session. And thank you so much for joining so early at Northwestern or Atlanta. I don't know where you are now. But yeah. Thanks so much, Benjamin, and see you next at the breakout session.
So let's move on to our next presenter, that is Janni Munk Christensen from University of Vienna, that will present novel eukaryote bacteria interactions in activated sludge.
Hello everyone, my name is Janni and I'm a postdoc at the Division of Microbial Ecology at the University of Vienna in the group of Mickey Wagner. And today I'm going to show you a glimpse into our current work on Project Novel Eukaryote Bacteria Interactions in Activated Sludge. Interactions is quite a broad topic and can mean many things, ranging from co-occurrence in the samples to understanding positive or negative effects on either the host or the microbe. For activated sludge, historically many eukaryote bacteria interactions related to the treatment process has been described. Especially before it became common to sequence the microbes, certain groups like protozoa and metazoa has been indicators of the treatment's performance. In general, eukaryotes has an effect on the sludge mass simply by eating bacteria in general, but also by being selective grazers on bacteria related to, for example, bulking or pathogenic bacteria, improving the sedimentation and ultimately the effluent quality. Historically, larger eukaryotes has been studied with microscopy method, and identification of them has been based on morphology. But it's hard to transfer this morphology-based knowledge and combine it with the recent modern molecular methods, as first of all, it's hard to do the identification, but when you, for example, do fish on a eukaryote, it's also very hard to keep the structure intact. And then you have a bad starting point if you want to study interactions with eukaryotes and bacteria in a complex sample. Also, identification by sequencing is a challenge for the eukaryotes, as we here have really far from complete databases. Here to the right, you see the expansion of sequences in the Silva database during the years. And even if we now have 10 million sequences there in the database, only 600,000 of them are actually from eukaryotes. And if you then look at the non-redundant database, the most recent version, it's only down to about 60,000 eukaryote sequences. And then you have to keep in mind that these are from all ecosystems and not necessarily from wastewater treatment plants. And also, if you look at it in a fish perspective, the newest fish probe that we use to target the eukaryotes are also from 2002, where we had much less knowledge about the diversity. So the aim of this study is to link sequence to morphology for eukaryotes in the activated sludge. And here we will, from one grab sample in a wastewater treatment plant, establish a small ecosystem-specific database where we, with primer-free methods, would get both the 16S and the 18S sequence. And with that, we will design fish probes towards the eukaryotes and then image them. And on the side, we will catch single eukaryotes from activated sludge and then do amplicon sequencing on them to obtain a list of bacteria that could be potentially associated with them. And when we got that, we would like to study further the interactions with bacteria, such as symbiotic interactions or food. To obtain the 18S sequences, we used some methods that I will just briefly mention here. We retrieved the SSU sequences by both an RNA approach, where we converted RNA to cDNA and sequenced it on a PacBio platform, but we also sequenced DNA from the sample using nanopore sequencing with Q20 chemistry. We generated quite some data with both of the methods, and then we took our data and hooked out the rRNAs to generate a tiny database with only our sequences. From this amount of data, we ended up having about 7,500 99% clusters. Here, most of them were bacteria, but we also had 120 eukaryotes. And I designed fish probes for some of the eukaryotes as well. These 120 sequence clusters are from very different eukaryotes, representing much of the diversity that you would expect to find in a wastewater treatment plant. This with both amoeba, ciliates, fungi, algae, and metazoa as the examples. I have designed fish probes to target many of the groups, and today I will focus further on this ciliate here that I have highlighted in red. And for this, we have optimized the fixation protocol for fish, so we can use fish and target it in the original sample. Here to the left, you see the fish picture of this specific ciliate attached to a small sludge flock. And we also managed to catch this type of ciliate and get it amplicon sequenced to obtain the bacterial profile, which you see here on the right. You can see that compared to the supernatant and the sludge fraction, specific bacteria from the Microbiota and Peteschi bacteria were much more abundant in the ciliate itself than in the control. Just as a take-home message, we have found here that it's now possible to use a primer-free sequencing method to retrieve 18S sequences from wastewater treatment plants. And we have used it in this study to retrieve 120 different 18S sequences from a single wastewater treatment plant. And then we have, from these sequences, established a small database and designed fish probes towards some of the eukaryotes. Then we have used fish to target novel eukaryotes in the activated sludge, and then combining this with new optimized fish methods, we can finally link the sequence to morphology and utilize some of the older knowledge about the eukaryotes. Then, by catching and sequencing single eukaryotes in the sludge, we can get a list of the bacteria that are potentially related to the eukaryotes and have interactions and point us towards which organisms that would be really interesting to study. In this case, like the Microbiota and Peteschi bacteria. So here for my acknowledgement slides, I will first of all thank the people at Dome and my group leader, Mickey Wagner, for making all of this possible, and the sequencing facilities that have helped out a lot as well, and the people at the wastewater treatment plant in Klosterneuburg. And thank you very much for listening.
Congratulations, Jani, and nice to see that you are in Vienna now, while we were sharing office in Aalborg. Are you online? And if you are online, you can unmute your microphone and share your video. And is there a question in the chat, Sergio? A question in the room? Yes. So, Samar Peter, you can come to the standing microphone. It should be activated. Yes.
Just wondering, what would be your biggest takeaway for us in the audience on the difference between doing fish for bacteria versus, this method that you've applied here? I will, of course, read the methods part of your paper, but just before that, if you can have a few, pointers for us.
Yeah, so that was actually a good question. I personally come from the side of bacteria, and I've done fish on lots of wastewater treatment plant samples, only focusing on the bacteria. But what surprised me the most was actually the methods that we use now to fix these samples. They actually, when you look at them, you only see the bacteria, but it's because the method facilitates the bacteria being there. Actually, many of the eukaryotes, for example, the ciliates, they simply change morphology, or they just burst out. So there's actually not many traces of them left, and they do not handle these types of, for example, ethanol storage and drying very well. And that was what surprised me the most.
Thank you so much, Jenny. And we can continue this discussion in the breakout session. And indeed, combining microbial ecologic data from eukaryotes and prokaryotes is not always very straightforward. Unlikely fish can give a solution, hopefully. See you next in the breakout session. Thank you, Jenny, again. Sergio.
Now we are going to move to Boom Q Kim from University of Notre Dame that will present effect of protozoan predation on the microbial community structure of nitrifying MABRs.
Hello everyone. Today I am going to present effects of protozoan predation on the microbial community structure of nitrifying MABRs. MABR use canal divisional biofilm, where highest bacterial growth rate will happen in the middle of biofilm, because in this region, both electron donor and acceptor are not limited. However, outside of this box, either electron donor or acceptor will be limited, and the bacterial growth rate will be minimized. Since protozoa is aerobic microorganism, so their predation rate will be higher near the membrane area. So therefore, in this region, predation rate will overpass the bacterial growth rate, which causing the production of internal voids. So the previous research with heterotopic MABRs, predation create internal void within this, near this membrane area. And then following research showed that this internal void weakened the mechanical stability of biofilm, which caused biofilm slapping. However, most MABRs are used for nitrification, and the effect of predation in nitrifying MABRs is not studied yet. Usually, nitrifying microorganisms produce dense biofilm, so there's a possibility they are more resistant to predation than heterotopic biofilms. Also, AOB form dense cluster than NOB, and protozoa show size-selective grazing. So it means protozoa might consume NOB more than AOB, so which might cause the bacterial community change in nitrifying biofilms. Therefore, the objective of this study is to determine effects of predation on nitrifying MABRs, such as internal voids and mechanical behavior and nitrification performance and microbial community structure. We grow 95 biofilms on flaccid silicon membrane with ammonium medium. Then we assess 95 MABRs with and without protozoan inhibitor, which is cyclohexamide, such as biofilm structure, microbial community, and nitrification performance. We use optical coherence tomography to image biofilm structures such as void and thickness, Qpcr for quantifying AOB and NOB, FISH and confocal laser scanning microscopy for in situ visualization of NOB and AOB in 95 biofilms, rheometry to measure mechanical stability of biofilms. Lastly, digital droplet PCR for quantifying the protozoa ratio in biofilms. Result and discussion performance. First of all, predation decreased biofilm thickness and nitrification fluxes. In this figure, the two bar graph on the right is for suppressed condition. The graph on the left is for control condition. Both nitrification flux and biofilm thickness was decreased by the presence of predation. Predation also led to nitrite accumulation. So for the control condition, nitrite was accumulated, as you can see here, but the suppressed condition, nitrate was produced. It means predation had a greater effect on NOB than AOB. What makes predation has greater effect on NOB than AOB? First explanation is because of their growth rate differences. Bacterial growth rate and predation rate is a balance each other. If there's more growth rate than predation rate, bacteria can become dominant. If there's on the other hand, if predation rate will be much higher than bacterial growth rate, there will be disappear in the end. Based on their growth kinetic, AOB more competitive than NOB when oxygen is limited. So they have a higher chance to survive by predation. Another explanation based on cluster size. Protozoa shows size selective grazing, and based on the research, AOB cluster size is greater than NOB cluster size, so they have a more resistance to predation, so they can survive more. So it makes predation has a greater effect on NOB than AOB. Qpcr analysis also confirmed that protozoa has a greater effect on NOB than AOB because suppressed condition has a higher amount of NOB than control condition. This is the FISH image of AOB for the control condition on the left, for the suppressed condition on the right. Based on this FISH image, we can say that predation of AOB does not seem significant. This is FISH image of NOB for the control condition on the left, for the suppressed condition on the right. Based on this image, you can say that predation of NOB is significant. Then we compare the FISH image of AOB and NOB for the suppressed condition, so there is no effect of predation in these two images. As you can see here, the cluster size of AOB is greater than cluster size of NOB, which is consistent to the previous research. Result and discussion for biofilm structure stability. Okay. First of all, predation increased internal void from 54% to 69%. And then this increased void weakened the mechanical stability, so both storage and loss modulus was impaired by the presence of predation. Therefore, predation increased biofilm sloughing from 4% to 18%. Lastly, digital droplet PCR analysis showed that there was a higher amount of protozoa for the control condition than the suppressed condition. It proves that all of the result of this study was made by the protozoa. In conclusions, predation decreases nitrification fluxes and preferentially suppress NOB, which promotes nitration in MABRs. Lastly, it increases internal voids and weakens the structural stability of nitrifying biofilms. Thank you for listening to my presentation.
Thank you very much, BoomQ. Are you online as well?
Yes, I am.
Yeah, thank you. Is there a question in the chat, Sergio? Question in the platform here on site? Yes, Bart, you can go to the microphone, please. Thank you. Question from Bart Smets from DTU. Eric, can you put the microphone of Bart higher? Thanks. It should work as well. I think that.
Nice presentation, and it's always beautiful to see those protozoa. And I wonder, it seems a little bit counterintuitive for the protozoa to do what they do, because the biofilm gets sheared off and they disappear and they no longer have their bacteria to graze on. So I'm always wondering whether this is an artifact of the system, or whether this has actually been observed in larger MABRs as well. Do you have any idea about how commonly this is taking place, grazing of these protozoa inside of MABRs, resulting in loss of biofilm structure stability?
And actually, in the real plant MABR system, we found a higher level of organism, like other than protozoa, such as nematode or like more bigger worm. So I think the presence of eukaryote microorganism in MABR system is very common. But in my system, since it is small reactor, we couldn't find the like bigger size of worm, but we saw the presence of protozoa, and then I studied that in my reactor. Yep.
Okay, thank you.
Thank you.
Thank you very much, Bart and BoomQ. So let's continue to the next speaker, and please stay online for the breakout session that we will start in 15 minutes. See you later.
So we are going to move to the last speaker of the session, that is Yevhen Miskevich from KAUST, and he's going to present live online, so you can start sharing your screen. And remember, after you start sharing your screen, please go back to the website to start the camera, because it turns off, so then we can see you while you present.
Yes, thank you very much. Good morning, everyone. I'll start sharing my screen now. And hopefully you can see it and hear me well. If something goes wrong, just don't hesitate to tell me. We can fix it. So you can see the presentation now, right?
Yeah, everything is okay.
Yes, thank you very much. So good morning again, everyone. My name is Yevhen Miskevich.
Full screen, if you want.
Yeah, this is a full screen now.
Can you launch the presentation mode?
Yep.
It's on your computer that we should do that.
Right now. Good now, right? Okay, so
you can do like that. It's fine as well. I think we can see.
So yeah, my name is Yevhen Miskevich, and I'm a PhD student in Professor Peng Hong's group in King Abdullah University of Science and Technology, Saudi Arabia. And today I'm going to present you with an alternative chemical-free approach for cleaning membranes from anaerobic bioreactors. Water scarcity is a global issue nowadays, and in order to alleviate this challenge, we can tap into clean treated wastewater. Most of the wastewater treatment plants nowadays are using activated sludge processes in order to treat the wastewater. Despite being efficient in terms of ammonia and CO2 removal and having an opportunity to subsequently recover energy from sludge, this technology still has some problems, as high energy consumption for aeration and also a huge area usage. In order to decrease their footprint, some wastewater treatment plants that are using activated sludge processes are being now modified with membranes. But still, this technology still remains really energy intensive, and in fact, it even increases the energy consumption. On the other hand, there is an anaerobic membrane bioreactor technology which utilizes absolutely different set of microorganisms that are able to ferment the organics from the wastewater directly to produce methane. Also, the elimination of the need for aeration means that this technology uses much less energy compared to the conventional processes. Additionally, the sludge production is much lower, and the effluent can be used as a liquid fertilizer later. But as any membrane technology,
then just a question: are you still on your title slide? Because we see your title slide now. So
no, I'm not on my title slide. I think there is some problem with.
No, the thing is that in your PowerPoint you have not launched the presentation. Now we can see it.
Okay, I'm sorry. I think maybe there is a problem with translating the slides to you. Maybe it's better just to start the video, because I have a pre-recorded video. If it's possible, please, just not to waste time.
Can you just try to, with your arrow to go down to the next slide on the left? If you go down.
Okay, so now it's working.
Do like that. It's fine. It goes very well now. Yeah, it's better.
Okay, yeah. I'm so sorry. So yeah, I was telling a little bit about conventional treatments and anaerobic membrane bioreactors and why are we sticking to this technology nowadays, like in our lab. And I was saying that it, as any membrane technology, it has the problem of membrane fouling. In order to mitigate this problem, the most commonly used solution is a chemical treatment. Basically, it is a use of sodium hypochlorite and citric acid. Despite being efficient against biofouling and economically feasible, this technology still has some cons. Among them, there is a toxicity for the environment, hazardous material formation, or it can also affect the membrane integrity, and some bacteria, as in case of Acinetobacter, are strongly resistant to chemical treatment, which will force us to increase the concentration. That's why there is a need for another cleaning procedure. We are offering to use the combination of bacteriophages and UV. Bacteriophages themselves are viruses that are using bacterial cells as hosts to propagate in order to continue their life cycle. Despite the efficiency of bacteriophages were shown in single species or simple multi-species biofilms, still their efficiency against complex biofilms remains to be pretty low because of the problem of penetration before encountering and infecting their hosts. That's why we have hypothesized that combining bacteriophage treatment with UV will improve the overall efficiency of the cleaning. Additionally, bacteriophage can be operated in two cycles, lytic cycle and lithogenic cycle. In lithogenic mode, bacteriophages are used to introduce new genetic material inside the bacterial host, but in lytic cycle, it uses the host to reproduce, thus killing the bacteria. One of the factors that helps bacteriophages to shift from one cycle to another one is UVC. So another assumption that was made is that UV can trigger bacteriophages to enter the lytic mode and then effectively kill the bacteria. So this research had two main objectives. The first one was to analyze the effectiveness of combination of UV and bacteriophage treatment to reduce anaerobic membrane biofouling. And the second one was to develop and implement a system to perform in situ cleaning. So let's talk about the first one. For the experiment itself, we decided to have 4 treatment procedures. The first one was control, which represents just untreated membrane. Second one was phages, which represents membrane exposure to bacteriophage cocktail, which is consistent of Acinetobacter modestus, Acinetobacter juni, and Acinetobacter sohiensis phages. The use of specifically these phages were due to the fact that Acinetobacter species themselves were previously shown by our group to be one of the core bacteria groups selectively formed in anaerobic biofilm. The third treatment was UVC alone, which represents membrane exposure to UVC, and the fourth one was bacteriophages and UV combination. So the overall setup was that 3 membranes were connected in parallel to the attached gross bioreactor configuration, which was fed with real wastewater with COD levels of 750 milligram per liter. The transmembrane pressure was monitored till it reached 20, 40, and 60 kilopascal, which represents low, subcritical, and critical biofouling. Also, our research has shown that in all 3 levels of biofouling, bacteriophage-only treatment was not really efficient compared to the control. In the meantime, UV showed statistical difference compared to control, and it was even more enhanced when combined with bacteriophage treatment for subcritical and critically fouled membranes. For low biofouling levels of membrane, there was no statistical difference between UV alone and UV plus bacteriophage treatments. We came up with the conclusion that it might be just to the fact that there was not enough bacteriophage host for them to propagate and properly do the cleaning. It can also be proven by the fact that for polysaccharide concentration decrease, there was no statistical difference between bacteriophage treatment and control for itself. But for subcritically and critically fouled membranes, we can see that there was statistical difference, and UV-only treatment showed also high efficiency in terms of polysaccharides removal. And again, it was even more enhanced by combining these 2 treatments. Also, what we have done, we have extracted RNA from the membrane biofilm that was harvested at 40 kilopascal transmembrane pressure and then treated with 4 different methods. Then we transcribed the RNA into cDNA and amplified for total 16 srRNA genes for amplicon-based high-throughput sequencing, which allowed us to see the changes in active microbial community. It was also seen that instead of just affecting Acinetobacter species, as the phages were Acinetobacter specific, there was a general reduction in many microbial populations. Among them, there were genera that included both gram-positive and gram-negative bacteria. So combining all the knowledge that we had, we came up with the main cleaning mechanism that UV kills the bacteria and loosens the matrix. Then the bacteriophages are able to penetrate inside the biofilm matrix and infect Acinetobacter bacteria, which determinantly impacts its ability to form the biofilm. Thus far, we have shown the efficiency of our system, of our cleaning in the invasive manner, which means that we have to remove the membrane from the case and then apply the treatment. This is pretty common experience nowadays, even for big wastewater treatment plants, where the membrane is extracted and soaked in cleaning reagent for overnight or even longer. But that means that the system stays offline, so it's not operating during all this time. So that's why we wanted to develop and implement the system that will allow us to do the cleaning in situ, hence maybe shortening the time that the system should go offline. For this, we have modified our reactor setup. There were only 2 membranes used for it. It was treatment, and the second one was used as a control. Also, we have, I'm sorry, this slide, it was animated. So we have modified our membrane cassette with 4mm quartz glass for it to be penetrable for the UV. And then we have also developed a protocol just summarizing all the knowledge we had before. You can see it on your screens right now. So we've applied UV, and when the membrane reached 40 kilopascal of transmembrane pressure, we detached it from the system and applied UV and bacteriophage treatments. Total UV radiance was 200 millijoule per centimeter square, and bacteriophage cocktail exposure time was 6 hours. Then the backflush one was done thrice, and then discarded through the waste stream before it putting this module back to the system and allowing the operation again. And during all the time of the experiment, we have monitored transmembrane pressure growth and water quality. Also, I forgot to say that the control itself, just the control membrane, it was exposed to soaking for the same amount of time in milky water and to the backflush, but it has never been exposed to UV treatment and bacteriophage. So on this slide, you can see the transmembrane pressure regrows after the first cleaning. And it can be seen that the time needed for transmembrane pressure to start regrowing for untreated membrane that is shown in blue was much shorter compared to the treated one. And also it took a shorter time for it to reach the desired transmembrane pressure levels. But as soon as the initial attachment occurs, the transmembrane pressure regrowth curve slope remains to be approximately the same, which means that the treatment itself has an impact on the initial biofilm colonization, but not on the subsequent formation of it. And we can also see that after the fourth round of cleaning, the transmembrane pressure didn't drop to 0, which might indicate that the irreversible fouling has reached the point when it's at this point we have to replace the membrane. Also, to estimate the total cell number, we've used the flow cytometry for both control and treated sample, and seeing that the total cell number was always higher for control, it can be said that slower membrane fouling observed on the treated membrane compared to control was due to the lower total cell number with every cleaning cycle. And in the end, summing up, I would like to say that isolated Acinetobacter-specific bacteriophages complemented with UVC has shown a potential as a new NMBR membrane cleaning method. Secondly, the combination of UV and bacteriophage cleanings outperformed UV-only and bacteriophage-only treatments in cleaning anaerobic membranes. The maximum efficiency of the mixed treatment occurred at medium fouling, and we have always also demonstrated that efficiency of the in situ cleaning approach. So thank you very much for your attention. And if you have any questions, I would be really glad to answer them.
Thank you very much, Jeven. And you were the first speaker online, but I think it went very well. I got sorry for this screen issue.
Yeah, I'm so sorry. I didn't know that it would be there.
We got all the messages. It went very well. Are there some questions in the chat in the room? Online on site, Mark van Loosrecht from TU Delft.
Also got a bit puzzled because Acinetobacter is a strict aerobic organism, and you're working in an anaerobic membrane bioreactor. What is this organism doing there, or did I miss anything on the setup of your reactor or of the development of Acinetobacter microbiology?
No, yeah, that's fine. Acinetobacter itself is a facultative anaerobic microorganism, so it can still survive in the anaerobic conditions. So that's why it was there in the reactor, and it was shown before we had the research that showed different groups results that all of them had Acinetobacter being as a core microorganism forming the biofilm. It was always deviating from the neutral model, assembly model.
Yeah, but not in the regular descriptions, Acinetobacter is described as a strict aerobic organism, so it can survive anaerobic, but it has not been described as being fermentative or growing under anaerobic conditions.
But
unless I missed some recent papers, but
yeah, it's still growing, and we were not really interested in the microbial community inside the reactors. We were interested in the microbial community on the membrane, so on the membrane biofilm, because the problem is the membrane biofouling. That's why we were interested what are the core bacteria forming the biofilm. And Acinetobacter and Methanobac, two of these OTUs, were always deviating from the neutral assembly model. So that's why we came up with the conclusion that we need to target exactly Acinetobacter.
Thank you, Jeven. Are there some questions in the chat? So please, online participants, I think there's a problem with raising the hand. We cannot see it when you raise hand. So really write your question in the Q&A or in the chat. Maybe a quick question to Jeven. What is the impact of UV on phages?
UV also has an impact on phages. That's why if we go back to I'll just start the demonstration again. Here. So when we go back to our treatment, you can see that we left the bacteriophages working for 5 hours. I said that the total bacteriophage exposure, like membrane exposure to bacteriophage, was 6 hours. So 5 hours, the bacteriophage has been doing its job. So firstly, we've exposed the membrane to UV, as I said, to disrupt the membrane integrity, to help the bacteriophages to penetrate inside. Then we added bacteriophages and left it working for 5 hours. And only then we applied UVC again, because bacteriophages themselves are also sensitive to UV. So if you do the exposure of bacteriophages in the very beginning, we've tried different permutations of our treatment. And it turned out that if you expose the bacteriophages in the very beginning to the whole UV period, like 6 minutes, there would be no effect from the bacteriophages. They are also slowly degrading. Their concentration is decreasing.
Yeah. So
there was
David,
some reaction online. So thank you very much, Yevhen.
Yeah.
So congratulations again for your presentation and thanks for your contribution. We will move next to a breakout session, let's say with all participants together to bridge all the talks together around this topic on microbial ecology in the water sector. I suggest we do a very short 2-minute sanitary break and refreshing, stand up, and we are back into 3 minutes and we will have 20-25 minutes of interaction then. So 2 minutes refreshing break and we are back.
ja, terminato la
wondering how he gets that. Was able to get that description. Well, the only point is that if we have a membrane and it's open to the air, that's actually fusing back into the membrane. But putting up the membrane means that the whole setup will work. Or the classification of the genome is not correct. Yeah, the features can go because looking is way through. Yeah, no, but that's the point. And it obviously didn't do that because that quickly does not check it. Find anything. So either the experimental setup is not right or just the classification. Yeah, no, that's. You know where? Yeah. That's a mistake. Except for the living conditions, the rest is okay. Yeah, when is it we don't move out here? Yeah, that's straight. To get it off the fly there.
Yeah, I think I was going to say that. I would like to use your for, yeah.
No, well, it's Singapore.
That's true.
But still, it's not a lot.
Is that water?
Yeah.
So,
You have to do that.
I know you do. I didn't know. Figure out
Yeah, it's also just asking around.
Eric, we will start the breakout session. So if you can also display the room again, this would be great. Pardon? Yes, it would be great. And we can give you a microphone.
Yeah, no, so it's like the building.
You can use the microphone there. You have to press the button so it becomes red. Yeah. So we are still in the same session as before, session block 1, microbial ecology, and we just need to be broadcasted again to the participants. And if all the speakers would join online, this would be great. So if you can show your video, all the speakers, so meaning Alaida is next to me here on site. So Susan, Benjamin, Jani, Bumq, and Yevan. If you can share your video, this would be great. And so we will test this breakout session. It's the first of the conference, but the aim is during these 20 minutes, we discuss about microbial ecology and the water sector. If there are some urgent questions, we can come back to them. We saw sometimes that some of you try to ask a question and were not able to do, so we'll give you the word as well. But maybe to make a summary of this session, what was interesting is that when we speak about microbial ecology, it's not only about bacteria. So in 90% of the cases, we focus on bacteria. They are the eukaryotes, they are the viruses, whether viruses or phages are microbes. It's not the question that we want to address here, but there are all these different types of biological agents that we have to measure in sludges, and there are different ways to measure them. So we have seen a dashboard with Alaida on amplicon sequencing to reach the operators. We have seen with Jani how we can combine 16S RNA, not the DNA, but the RNA sequencing directly to then design fish probes to target eukaryotes and bacteria, and to see this novel interaction or interactions in activated sludge. And then there are different types of effects that we can have in these sludges. So chemicals, as Susan has presented, like ozone, can have an effect on the microbial communities. Eukaryotes via predation can have an effect. UV, so a physical process, can have an effect, and phages as well. And then when we speak about eukaryotes, so Benjamin spoke about this autonomous in situ monitoring of microalgae. So maybe to discuss with the different speakers now on the audience on site online, is there a way to bridge all these different thematics that we have seen in this session? Could we end up, for instance, in the end with an autonomous in situ monitoring of activated sludges, if we start from the presentation of Benjamin on microalgae, and to have quasi this autonomous system at the wastewater treatment plant level to implement the dashboard of Alaida? So if I ask you, Alaida, first, and you have to unmute your microphone, what would you think about this idea?
I think, first of all, it's a great idea. I think everybody would love to have it. Not sure we're there yet, but obviously being able to combine all levels of the microbiome that are present, that would already be amazing, that we don't only focus on the microbial aspects, but also include the other ones as well. I think that would be a first step, maybe offline. But in the end, I think if you ask anybody who's involved with monitoring any kind of process, of course, you want it to be autonomous, you want it to be online, and you want it just to give generate data without you having to do anything for it, I suppose. But inherently very tricky, I would say. But yeah, something to look forward to, I hope.
If you look at your, if we look at your dashboard, so you essentially targeted bacteria, is there a way to directly include with amplicon sequencing both eukaryotes and prokaryotes in the same graph?
Yeah, but we don't do amplicon sequencing. So yes, we look at the total DNA. So the information is there. But as was already mentioned, the databases for eukaryotes is still rather lacking. So we haven't actually looked at the data, but that's one of the things that can easily be included if you just look at total DNA instead of doing amplicon sequencing. I think that would be a very important first step.
And combining the two types of data set in one single graph, is it possible, or how can we do that? Because if you think about nematodes, for instance, there are so many different cells in one organism, so it's multicellular.
Yeah.
Is the relative abundance of a nematode the same as the relative abundance of bacteria?
I think you'd have to correct for it, but there's extremely intelligent people that know how many cells they have and can correct for that and then visualize it, correct it for the number of cells they have. Just like we do often for Qpcr, you correct for the number of copies they have with them. So I think, once again, it's probably more of an IT challenge than a scientific challenge in that respect.
Jani, do you agree with this statement from Aleida? Because I would think that fish can also help in this direction to quantify relative abundance of bacteria, so unicellular and multicellular organisms as well. So I don't know if you tried beyond ciliates also with nematodes or other types of higher organisms to target them by fish.
Yeah, it is definitely a thing with, as was also mentioned, that we don't really have the super big overview of them to design the fish probes yet, because there is not many sequences in the database. And the sequences that are there, some groups are very well studied, but it has often been focused on few narrow groups. I see that fish can definitely be helpful in maybe answering some of the questions that we cannot necessarily see only with the sequencing. So if we the two techniques, with, as I see it, really complements each other and do not necessarily have to compete over this, and it can add very different information, both of them.
Yeah, indeed, the aggregation of the method is certainly the key, and the research question or investigation question for practice is the key to select for methods. Then is there just a gold standard that can be helpful for everything? I don't think so, and we need to aggregate all these methods. If you look in the chapter of Trina and Per about microbial ecology methods, you see that there are 30 or 35 methods that can be used in this context. Mark has a follow-up, please. Yeah.
Yeah, just for the discussion, because you just asked Aleida about, is it useful to have an online in situ measurement of the microbiome? And we can put a lot of effort on that and get that done, but I'm afraid that it's overdoing the job. The sludge age in many installations 10 to 20 days, which meaning the system is not changing every hour. In the microbiome, in the chemometric parameters it is, but in the microbiome it's not. And I think it would be much more useful to spend our time not to intensify our frequency of data with all kinds of tools, etc., but try to spend our time to interpret the data. And so I'm very much against this online goal of the microbiology.
Yeah, and this is definitely what we speak about often about, maybe measuring less, but getting more information out of the data. And maybe, Aleida, what will be your experience with the water boards, with this type of dashboard and potentially online measurement? There are also other groups that want to go at line with sequencing. Is it about the sequencing per se, or is it about the information that we can get to operate, design, control?
I totally agree with Mark that, of course, the interpretation side and knowing what to do with the information you're getting is exceedingly important. On the other hand, I do believe there would be an added value to be able to monitor the wastewater that's coming in on a more online perspective. to see if there's anything in there that's going to give you problems further along the line. So before you're seeing the effect in your sludge, to actually monitor the wastewater that's coming into your plant. In that respect, if you can see an effect already on your microbiome, it could be very valuable. But that's a different application than using it to actually monitor the activated sludge.
But is that a microbial analysis or a chemometric
Also
analysis?
microbial.
But can you indicate what in the influent would be responsible on the microbiome level for what you could measure there on microbiome level, which could influence the treatment operation?
Yeah, I think especially for the nitrification, if you're seeing that you're, especially in plants that are inherently receiving a lot of industrial wastewater, one of the things is to see if the nitrification is being inhibited. And that's one of the areas that would be interesting to, instead of taking a sample to a lab to perform a nitrification test, to be able to monitoring the activity of the nitrification biomass online to see if it's being inhibited, if it's having issues.
But that's not monitoring the influent or doing a toxicity measurement on the
It's
influent.
more towards toxicity, absolutely.
Right, so
but I
that's-
think you could still use the same technologies and the same methods to do that.
Oh.
But yeah, no, it's a fair point.
The toxicology measurement, that I agree, by the way. But I'm not sure whether that should be a microbiome measurement.
Yeah.
That's in general a question, because you see all these presentations, you report data from sequencing. Now, we all know that the sequencing and the interpretation, the sequence itself, the data analysis, and the interpretation of that has biases. And I haven't seen anyone using a control measurement for your results. So if you see there is some organism or some organism dominant, what would be also for the online monitoring, proper control measurements to be sure that what you analyze is really what's going on and not an artifact of the DNA sequencing or the database interpretation.
Suzanne, would you have a follow-up on what Mark was asking here, since you work on the effect of ozonation on activated sludges? So who is your opinion about what Mark mentioned just now?
Well, I totally agree with Mark, but I think in our case, we just were so lucky that we had a control line, so we could simply just compare with all the biases, but in both lines. Yeah, but of course, he's right. There is a lot of biases, but we are aware of them when we use these methods.
And so to which extent the higher resolution that you got from amplicon sequencing was useful to make a statement about the effect of ozone on biomass? So should we look at the biomass level per se? Okay, the biomass decreases in terms of concentration, or is it an effect directly on some specific population? So how should we go further with this data to, for instance, investigate the effect of ozonation therapies or bacteriophages therapies, if we link to what Jeven spoke, or with eukaryote therapies or ciliate therapies with BunQ and the others?
I don't know. I think we need to combine them also with the process data to look at the performance. I don't know. Any input?
Yeah, colleagues behind can also input. And BunQ, Jeven, do you have any follow-up on this? And Benjamin? How can we make a difference by getting all this different information about bacteria, eukaryotes, viruses, or phages in these ecosystems? How can we tackle this complexity? Is it to sequence more, or is it to get more specific information on specific populations? Maybe BunQ?
I'm sorry, my microphone is like...
So we were discussing about should we sequence more? Do we need more data, or do we need more targeted measurement on specific cases? For instance, in your biofilms with the interaction, I think it was with protozoans here. What do you need as analytical methods to better understand your system? Or what do you need less, maybe, as well?
I think in my case, I specifically target
protozoa. So I don't know. And then I want to see the effect of protozoa on the nighttime biofilm. So in my case, specific targeting would be beneficial, but it depends on the purpose of the research. I think in some research you want to see the overview, big picture. So we want to see total DNA sequence. But if we are not interested in that, we don't need to do that all the time. That's my opinion.
Yeah, thanks. And Yevon and Benjamin, what would be your opinion on these points?
So if it's okay, I'll take a word. And for us, we are still working on this project. So this project is still ongoing. And right now we are going to analyze 16S rRNA amplicon sequencing data. So, and we also are going to analyze metatranscriptome data. So we want to see how our treatment is really affecting the bacterial community inside, and if it's applicable, because, you know, what we've already shown, it's still, you know, in real world, it's a little bit more tricky. In research, you can show the data, but to implement it in the real life, it's a little bit more tricky. Of course, we have to do the cost analysis. For our technology, there is some work to do the cost analysis, to compare chemical treatment, because unfortunately, chemical treatment is really economically feasible. It's cheap. So yeah, we have to do this one. And we also want to see how our in situ cleaning is affecting the microbial community on the membrane. So this is what we are going to work. This is what we have to pay attention to before saying something to implementing it in real life.
Short follow-up, how can you see something at the transcriptome level, meaning at gene expression level, about the effect of a bacteriophage on bacterial biomass?
Of course, we want to see what is the, you know, that 16S amplicon sequencing might be not that really, how to say it, precise. We want to see from both perspectives, because we have extracted RNA, so we are interested only in living community, so a live community on the biofilm. And we want to see how it changes, so how our treatment is influencing the community itself and its ability to form the biofilm, because you know that the biofilm itself, it's not only bacteria, it's also EPS, extracellular polymeric substances, that is polysaccharides, proteins, so a lot of different things. So we want to see what our bacteria can do there. So, and how can we mitigate this problem of biofouling itself? That's why, yeah, we want to see on what is our active community and what it can do.
Thanks. So I think it's important when you select a method that you think about what is the target that you want to analyze with the gene expression. Thanks, Yevon. And we come back to this autonomous system with Benjamin again. So you started this system, developed this system for surface water monitoring of algal blooms. How would you see that in a wastewater treatment process or drinking water treatment facility? How can we could we or could we not implement these technologies?
Yeah, that's a great question. I'm actually currently in my work pivoting away a little bit from surface water algal bloom monitoring towards wastewater treatment, specifically globally, but especially in certain parts of the US. There's an increased use of microalgae to remove nutrients from wastewater, and I hinted that in my presentation. So in cases where we're using eukaryotic organisms to help the wastewater, in this case, the microalgae are used post-secondary treatment to remove excess phosphorus. In these cases, doing imaging is a much faster way to get information on how the biomass is accumulating, any changes in community structure. Of course, doing something like sequencing will give you the best taxonomic resolution, but kind of as Mark was saying, we don't always need to be checking this every couple hours because these communities are relatively stable over time. But at least in these microalgal communities, the system has an SRT of about 3 or 4 days. So we need to get results usually every day. It's easier to do this if it's an autonomous system than having to have someone take samples every day and run them. But you know, generally we're looking for for changes on the orders of days, and whether it's like predators, grazers, rotifers, anything that's large enough to pick up on in these microscope images, I think there's a use case for this instrument to look for, anything. I mean, we can see bacteria as well. The taxonomic resolution is not so great. You can't really tell what kind of bacteria it is, just that bacteria is there. So in the case of, quantifying bacterial community structure, I still think sequencing-based methods are more appropriate. But yeah, I think for anything bigger than a bacteria, imaging is the way to go.
Thank you, Benjamin. I just have a question to Zeb. Will the session stop at 11:30 per se, or we can still continue with some questions to the participants? Are you online, Zeb? I just hope that the session does not close, because there were some burning questions as well from other presentations, and it's good that we can give the word to the participants.
Yeah, I saw a question in the Q&A from Ezequiel, so maybe you want to join the discussion and ask it yourself.
Can you guys hear me? Hey, can you guys hear me or not?
Yes.
Yes.
Okay, that's good. So what was the question? Oh, so the question is related to what Mark asked to Aleida earlier about frequency and getting that data, right? Getting deep into a few samples or measuring more frequently, right? And part of the work that I do tries to capture that natural variability and stochasticity that we often encounter in complex systems. And it's very, very hard to capture that with few samples. We actually need replicated systems. We need more samples if we want to capture this variability, right? And then there's more and more evidence that suggests that these communities can go to alternative stable states. And for that, we would potentially need more frequent sampling. So I would like to, with this view, ask the members of the panel, what do they think?
Aleida?
Yeah, no, I suppose it's also very much at what level you're looking at a system. I mean, if you think about it, at the moment, we're still searching very much of how, if we go back to how we're translating the science to go to practice, I suppose the practitioners also need to indicate how much detail they need and want to get from their system. And because we're still searching very much, as you say, with what different types of steady states are reached, how do you, which are the sort of key parameters to monitor in order to know what they're doing, I think it's going to have a natural evolution of in the first instance, trying to get more and more samples and more data. But hopefully towards the end, you know what the key parameters are. And this could be a physical or chemical a parameter that you monitor with a sensor, is very simple method, or a simple Qpcr analysis for one key parameter that is indicative for a whole system. Or it could be necessary to do more frequent and more autonomous and more online monitoring. But I suppose we don't really know yet where we're going to end up in the future. But moving towards finding out is a really important process. So I suppose at the moment, from science and from practice, you're looking at getting more data with perhaps the ambition of getting less, because more data costs more money generally. So in order for the taxpayers' money to be used for other things, perhaps it's worth eventually going towards less monitoring.
And this always brings us back to, should we do a respirometry test? And this is sufficient to know what is the nitrification activity, and it comes often in our discussion with Mark about that. Or should we go at higher resolution to monitor what happens in the communities with amplicon sequencing, metagenomics, metaomics, metaproteomics, metabolomics, etc. So the data should be processed as well. So if you make your respirometry test in 1 hour, you make data processing in 6 months. So there's also the time to get the information out of the data, and that all these skill sets should be developed. So also in our university curricula, we need to integrate much more these types of data processing as well to render it not high throughput but fast throughput as well. There is this balance to find, and we'll see in the next sessions today and tomorrow, notably where Ezequiel will speak about theoretical microbial ecology, that if you want to address stochasticity and immigration from influent, for instance, you need much more resolution data, so it depends on the question again. I think there was a question also from, I think, Kwe from the chat. Sergio.
No, Kwe's question. We have a question from David.
Pardon?
Hello. I have a question for Jeven about the bacteriophages. I find very interesting that you are trying different methodologies for preventing biofouling, but I was wondering if you have given a thought, apart from the techno-economic analysis and etc., the feasibility of the project, about the biosafety issues. Like by the fact that you are using bacteriophages, you are gonna release DNA from industrial scale wastewater treatment plants, in case it was translated to it. And Acinetobacter is known for being one of the potential hosts for resistances, antibiotic resistances. I'm bringing it a bit to my topic. So I was wondering if you have given a thought on what could happen with the DNA that's being released from the bacteria that you are killing in biofouling and in membranes. Thank you.
Yes, thank you very much for your question. Honestly saying, no, we were not considering the, let's say, drawbacks of this one. That there would be a DNA going out from the bacteria. But what I can say, if we apply our treatment, like as I said, bacteriophage and UV treatment, we know that UV itself causes the damages in DNA. So the bacteria that are exposed to UV are mostly not able to multiply, so to continue their life cycle. So that's why, yes, we will have some DNA being released to the environment, but still at the same time, we will have a lot of this DNA being exposed to UV, and that's why being damaged. So this is what I can say. But being honest, we have not discovered, like we have not thought about this in our project. Not yet. Maybe this is a good point to pay attention to. Thanks for your question.
And this bridge to the session, I think 7 on antimicrobial resistances, where David is speaking and other colleagues as well. I think also in your group in KAUST, you are also working on eDNA. So it can be a nice link actually to discuss with your colleague also on site. Sergio? Is there other questions online? So we are a bit over time and it's time to go to lunch. If there are some burning questions, we can take one or two last ones. Or from the audience. Yes, Mark, please.
I'd actually a quite direct question for Yanni. Did you compare the protozoa sequences you found in your system to the existing protozoa in the database, and how much overlap was there?
So yeah, thank you, Mark. Yes, we did somewhat compared, and I made a comparison with the Silva database, as these are not really included in the MIDAS project. Here I saw that many of the, we have the usual suspects like the model organisms like the algae chlorella, we have the Vorticella organism, and we have the Saccharomyces, and we have Candida, and these, for example, some amoeba sequences that are in the database, but it's like half and half, where you do have a representative there, and then it's somewhat similar, but you do not have this direct organism there. And that's what we often see from these 120 sequences.
Thanks, and maybe the connection to Aalborg then is about, will we see once MIDAS database of eukaryotic organisms, of activated sludge and digesters, Susan, and colleagues behind?
Yes, yes. I think they actually will be part of this big microflora danica project that Pia is having with Mass these days. That they are trying to do the add-on with the protozoa and the eukaryotic microbiota of Denmark. So it's coming.
Thank you very much. And I think this is the award for the end. So we need to aggregate this data in databases as well to make sense for the data that we will collect in the future. Thank you so much, Aleida, and all the speakers. So Susan, Benjamin, Jani, Bunkyu, and Yevan for your nice presentation. Very nice interaction now. And we have 1 hour around, or 50 minutes to take a break, a lunch, or an evening meal, or a morning meal, depending on your time zone. And we see each other, in 50 minutes for the official. No, sorry, I have to check the platform, the program again. Thanks, thanks, Sergio. Paper is still good on digital platform. So at 12:30 CET, there is the IWA MIWI Management Committee meeting. It is on a separate Zoom platform that was sent per email. At 12:30, there are the roundtable posted discussions in parallel to the MC meeting. So it means that you should be able to join online to discuss with each other, with multiple participants across all the posters of different topics, these 5 topics of posters. And the official speeches will start at 1:15 Central European time, with notably the chair and secretary of MIWI, so Bart Smets and Amit Pinto, and a representative from the IWA. So we will welcome the IWA president, Tom Mollenkopf, and Samuela Guida, who is the head of the engagement program and strategic program of Iowa. Let's see each other in 50 minutes further and have a good meal and refreshment. Thank you so much for this session this morning.
I wouldn't leave the back here. Are you closing this? Okay. Well, I have nothing. Yeah, I have nothing actually. My I forgot everything, my computer, everything at home. I will go through that 1 second.
So 1 second.
I will wait for 10 minutes. 10 minutes. You only need to 5, so it's literally 5 minutes.